conference. So this is section 4. The topic is AI in biomedical image. The moderator is Professor Kun Huang and myself. Actually, today we will have 4 excellent talks. If you have any question during talk, just follow what has happened in last section. So you can just make your questions in the chat box. And after the 4 excellent talk, so we will have a panel discussion, and 4 of the speakers will address and have a lot of discussion. So thank you so much. Firstly, it's my honor to introduce our first speaker, Dr. Tanvir Said Mohammed, as our first speaker. So she is the IBM Fellow and the Global Leader for Image AI in IBM Research. So she lead a lot of initiatives in IBM and worldwide in image AI. So she have published over 200 publications and filed over 100 issued patents. She has led many great, excellent conference in medical image and the image processing in general, like CVPR, ISBI, and the MICAI. So she will be the MICAI 2023 General Chair. She is also Fellow of IEEE and AIMBE Fellow. Thank you so much. Dr. Said Mohammed, please start your talk. Thank you.
Okay, thank you very much for introduction. And I'm happy to be here and talk about the work in chest X-rays that we've been doing as part of a Radiology Grand Challenge project. People can hear me okay?
Mm-hmm. Yes.
Okay. I am going to turn off the video so we save on bandwidth, and hopefully the conversation will be clearer. So this is the work that is done at the IBM's research labs in Almaden, California. So you can see a cool building over here that I haven't been to for a year and a half. Hopefully we'll be going there soon. So let's see. I just wanted to acknowledge my team that has contributed to this work over a number of years. The Radiology Grand Challenge project has been running for a number of years, and we just concluded it last year. And what I'm going to talk about is one of the major initiatives we took during that challenge. And as you will see from my presentation, that to pull off a challenge of that type, we need cooperation and collaboration from many different specialists, not only medical imaging researchers, but also deep involvement of clinicians, radiologists, and as well as general clinicians in our work, and software engineers and data scientists and statisticians and even regulatory personnel, as you'll see. So what is the Medical Film Radiology Grand Challenge? This was a project we started that helped kick off the field of AI in radiology. And if remember, we did an exhibit called the Eyes of Watson in 2016 at RSNA, which also helps get it exposed to radiologists from what used to be in medical imaging as a focus area. And since then, RSNA is holding an annual AI showcase, and SIM and other radiology focused conferences have picked it up as well. So the basic idea of the Radiology Grand Challenge was to produce cognitive assistants that could serve and augment radiologist workflows, and do it in such a way that they can be trusted and at a level that is at the level of an entry-level radiologist. So the products that came out from this work and all, I think you've already seen from IBM Watson Health Imaging has already learned some of the products that came from our research. But the challenge that we took on in this particular effort was to see if a machine could do a full preliminary read of a single modality, and which means going directly from an image to a preliminary report, and then show that the type of report that we generated is as good as what could be done by an entry-level radiologist. So now you may ask, what is the preliminary read problem? As I said, it is going from an image such as this to a report that documents the findings and the major structures, both positive and negative findings, as well as a declaration of normality from which potentially could be used for filtering as such. But look at the phrasing of the report that mentions the problems and in terms of where the problem is located as well, not just what the anomaly is. So we chose chest X-rays, as most of you know, it's the most common modality, and it's also from a commercial standpoint that makes sense given the prevalence of use the radiology practices don't make much money on chest X-rays. So having machines do much of the filtering would actually be welcomed is the current hypothesis. There's also a lot of error, the possibilities due to the low quality of the imaging and the cognitive biases that hinder the interpretation of these as such. So if we could produce a report automatically from looking at chest X-rays, if the machine could do that, and if that was a good quality report, it can help expedite workflow in many ways. We can see, sorry, I think, okay, so the idea being that it can be used at any steps in a clinical workflow, whether you are an imager and at the point of imaging, determining whether the quality of the imaging is adequate, and the reports would establish that, because technical assessment is one of the things that would be in the report. If it went into the PAC system and there was a criticality of a case, then you could reorder cases, so it helps prioritize important cases to be read ahead. If obviously if the preliminary report is already made, it in some sense disrupts the current dictation-driven workflow where the clinician has to dictate and sit and correct the report or outsource it and wait for that transcribed report to come back. So in this particular case, you would be able to get a preliminary report that a radiologist can quickly correct and then enter it into an EMR. So overall, it would expedite the clinical workflow is the expectation. So now let me just show you what we mean by doing a report. So given an image such as this, the system would go ahead, and this is a service that is running. You submit the image to this service. It analyzes the structures and then comes back with a finding report. And this is the capability we brought it to. And you can just select any image from your PAC system, any study, just upload it to the service, and you would get back a full-scale report. And you can see here the image quality is pretty often there is skew and so on, but still the findings were detected and technical assessment was made, viewpoints established and so on, and then the report is generated. So so you might say, why is this a grand challenge? So the grand challenge comes from the fact that the imaging that we are interpreting is, as we said, very poor quality as such, and a variety of it from various hospitals and settings. But the most important reason is that if you want to claim that the machine can actually produce a report that is at the level of the entry-level radiologist, it must be able to have a full coverage of findings. And that's the first difficulty, because we don't know how many findings are there in chest X-rays. The second is to go and do them as accurately as possible. But if you're saying it should be comparable to radiologists, how good are they in detecting? And we don't have any concrete data about them, since we don't know how many findings are there. Then the last step in reducing the report form is to be able to describe the finding. And so use terms such as these: calcified right hilar and mediastinal lymph nodes. So this not only describes the finding, but also where it is occurring, laterality, location, severity. All of these things need to be described as well. So overall, how do you judge the quality of a report, and how can you actually do formal comparisons? All of these makes it a grand challenge. So as you can see from the description, there is work to be done, not just on the machine side, but also actually establishing the benchmark itself on the radiologist side, where you need to be able to catalog all such findings, first of all, and gather benchmark data sets that are agreed upon by radiologists themselves, and the findings from them have been properly documented, so we can have a gold standard for judging as well. When once you have that, obviously you have a lot of work to be done on the machine side in terms of assembling training data sets with different prevalence distributions, labeling such images accurately so that you can build machine learning models and then develop some comparison studies as well. So this is all we had to do in order to conduct this project. And some of the many data science problems that we had to solve along the way, I'll point out, as this is a conference about AI and its use in biomedical imaging, many of the problems in data science also apply to us. And you have mentioned over here some of these problems that we'll take you through. And in fact, the coverage will cover most of these issues. So let's look at the first one, which is to catalog the full spectrum of findings. Now, as you know, the majority of the work in chest X-rays has been on describing mostly the 14 findings that were initially done in the NIH dataset. And a lot of the new datasets that have come out publicly available have basically gone after essentially the same set of findings. There has not been a very good systematic effort to catalog how many findings are there. So this is the effort we took on, and essentially the approach we took was to see how to catalog the findings by closely following the radiologist process. So you can see that a radiologist would first to see whether the image is ready for interpretation from a technical assessment standpoint, whether it is underexposed, overexposed, whether the viewpoint has included the majority of the regions that needed that were supposed to be evaluated and so on. Once we know what that it is a good image to interpret, it would also be important to know whether it is an AP or a PA chest X-ray, whether it is a portable setting, as this could then establish some biases if the patient is, if it's an AP chest X-ray, then this is probably an in-hospital setting, and that already sets up a bias for interpretation. And if it's an in-hospital image, then one of the tasks is also to see if there were any tubes and lines inserted and whether they were properly positioned, and if there were any artifacts, device artifacts such as sternotomy wires or pacemakers that need to be described as well. And then looking at the various anatomical regions, looking for abnormalities within them, see what are the implied diseases from them, such as pneumonia and so on, and then eventually generate a report. So this is the typical process a radiologist follows. I'm not sure if there are radiologists in the audience, but perhaps they can comment on that as well. But this is what our radiologist used as a guideline to help develop the algorithms as well to address each of these problems in the cataloging of findings. So overall, we went through an effort with many clinicians using both a top-down and a bottom-up process to generate a lexicon of that describes about 237 discrete findings. And you can see over here the catalog of this. This essentially is one of the largest set of core findings assembled, and they cover the various types of findings ranging from anatomical findings to tubes and lines to their placements and so on. Some of these data sets we've actually also released in open source. So you can see over here that the generation of the findings took both top-down knowledge from the clinicians and guidelines, but also bottom-up extraction by mining about 200,000 reports, where we used a tool that has an active learning capability, where you use self-supervised learning methods for encoding of text and use that for similar expansions of similar terms. So this is a tool called a domain learning assistant, which is used by clinicians to rapidly expand the vocabulary. Much of this work we have described in the EMEA publication last year. So based on that, we gave rise to a whole bunch of finding labels. So you hear some of them are documented here, and the radiologists in the audience can relate to many of these findings as such. So the next step was to go and acquire data and label such data sets for these findings. And obviously, when you want, the key considerations we'd have to do is to see that we're not just trained on one hospital data, but can actually do it across demographics. And to be able to understand the prevalence distributions of it and use them for evaluation purpose. So our data sets came from at least 3 different sources, and we set apart some data completely for testing that came from an international data set as well. And this is the data set from India. And you can see that the prevalence distributions and the way the partitioning of the data sets was done for training, testing, and validation maintain the prevalence distributions as such. Many of this MEMIC CXR database is now available also in open source, but at the time when we started, this was exposed to us through a consortium agreement. It has about 60,000 patients and about 450,000 plus images as such, whereas the NIH dataset came with about 100,000 images, but from 30,000 patients, but only a small subset of them we could use because the labels that are provided only covered 14 of them. So we ended up having to relabel this data set to a large extent for the full coverage of findings as such. So the next step was to label these data sets. So many of them now, there are labeling platforms. Back when we started, there weren't any good web-based labeling crowdsourcing platforms that could be used for this purpose. So we built our own such platform. However, using that platform and about 40 radiologists, we could only annotate about 30,000 images in 4 months. And this was, if we wanted to go up to 1 million images, this seemed like a tall order. So if you see many benchmark data sets collected, the number of images are also limited because of this, the manual labeling consideration. So we actually went ahead to develop an automated labeling method that allowed us to label this in an accurate fashion. It actually used a combination of machine learning with vocabulary driven and language modeling approaches to come up with an accurate description of the labels as such. And with that, the labeling task, once the algorithm was developed, the 500,000 images could be labeled with verification within a week. And the whole labeling process itself took only a few hours. So we evaluated this for purposes for core finding detection, and you can see that typical pipeline used for text analysis and the current accuracies for such things evaluated on test corpuses. This is also a work that we published in EMEA last year. The key features in here is the way the negations, negative findings were captured in the descriptions using pre and post negation rules as such. And the coverage for such findings you can see in comparison to the 14 X-ray findings and the current performance using F-scores for across the findings. You can take a look at this data set. So the next step was to actually not only just describe the core findings, which we were able to use using the chest X-ray lexicon we had developed. It was to look at the variants of the spoken phrases and to recognize through synonyms and so on. But if you want to now group these findings with their descriptions that describe the locality, laterality, and so on, then we need to go through a deeper language analysis as we go through those approaches. So in this particular case, the highlighted phrases are some things that we would want to be able to extract and associate with the finding. The predominant way to do this has been to use machine learning language generation approaches such as the transformer-based methods. But those approaches, as has been our experience, don't guarantee the clinical correctness, even if they can get natural language ways of generating. It was important to make sure that the findings that are generated from reports had to be clinically correct. So the labeling methods had to ensure that the finding descriptions that we are capturing are clinically correct as well. So the approach we use for that is to analyze a sentence and use the core findings to actually label the various elements. So for example, we know pneumonia is a disease, clear lungs is a description of a finding, and the lungs refers to an anatomy. So this information we can get from the lexicon if you look at a typical sentence from a report, and this is the companion reports to your current chest x-rays. Then we use a technique called phrasal grouping, which does a dependency parse tree analysis of the natural language sentence, and it successively groups these into forming larger and larger connected components that link core findings with the associated modifiers as well. And the modifiers themselves are also in our chest x-ray lexicon. So for a sentence such as this, we can actually generate a finding that it is an anatomical finding, and it is a negative finding. It's associated with a core, the description of the finding itself, and the one or more modifiers that refer to what section, what portion of the chest x-ray are we talking about, and it can also help describe the character and appearance of the finding as such. So here's another example. Given a sentence such as this, the lungs are normally inflated without evidence of focal airspace disease, pleural effusion, or pneumothorax. You can break it down into 4 sets of findings. There are 3 of them are anatomical findings. One of them is a disease, which is an airspace disease over here. Most of these in this case happen to be negative findings. But this type of detailed description of findings enables us to do a fine-grained label report generation that I'll talk soon. This work, by the way, at EMEA won the Best Paper Award last year. So from the fine-grained descriptions, you can see that it's easy to generate the labels. So given a finding such as this, it's an anatomical finding of atelectasis. It's in the lung, and it's in the left lung and in the basal region. You can issue a statement like left basal atelectasis. We actually analyzed the detection capability for such label extraction, accuracy for a number of reports in the Indiana collection again, and we find that the label extraction accuracy is less than 4% error, but in fact only less than 1% error in associating the relevant modifiers, but the majority of errors are still due to negation sense detection, which remains a challenge in text analytics. So with that, we now had to go to the next step of answering the original question we came, which is how many fine-grained findings are there? Well, the lexicon gave us the core findings of the 200 or so odd. If we were to now catalog the full spectrum of fine-grained findings for a machine to develop, you can see that there are many. In fact, once you use this pattern extraction methodology for about 200,000 reports, we found about 100,000 of these labels. But if you could essentially assemble and group them a little bit further with respect to the resolution at which you want to describe, you would still get about 1,700 or so findings. Once we analyze their descriptions and try to do a mapping of which of these findings, if we were to retain, we could cover about 80 to 85% of the set of chest x-rays. We were, after evaluation and curation, we got down to about 457 findings. So this is the current set of findings that, that we feel is sufficient to describe accurately the findings in chest x-rays and still generate a report that is of reasonable quality. So now we see that there is a much larger set of findings that had to be recognized by machines. But by using this methodology of fine-grained descriptions in that derivation of the machine learning labels itself, the report generation process could be made easier. So let's go ahead and now build the models as we go from such provided labeled images. So the current methodology is auto-labeling for images from their companion reports, and we can go directly to building the machine learning models. So the methodology we used for this, initially we tried to develop specialized deep learning models, thinking that it's better to divide up the problem and focus on each of these and do. So some of the machine learning models we built are models for normal abnormal discrimination, tubes and lines classification, technical assessments. Each of them can have their own applications as we go. But the interesting learning we had was that even if we break this down, the best performance can actually be obtained by putting them all together into one machine learning classifier. And so in the end, we ended up keeping one classifier itself for all the 457 labels, but do it in a coarse, fine fashion. So the architecture we adopted combines the good features of most modern machine learning networks for computer vision. So those that are pre-trained from VGG or ResNet 50 features were built into a multi-scale pyramidal networks, which give you multi-level feature extraction, so to say, and capturing the various abstractions and describing the local and global abnormalities through dilated blocks. So this also gave us to the second feature in there that allows you to describe it at various levels of resolution, the anomaly itself. And then finally, a nonlinear pooling technique we use, that instead of doing a flattening that typically happens before you go through a classifier, you do a feature expansion through a global square pooling before you do the flattening. This allows us to capture nonlinear relations between features produced by the feature maps as such. So the details of the architecture, for those who can follow this along, is shown over here. The methodology we use minimized the number of parameters you had to train as well, but even so, the images we were working with were 1024 by 2024 to begin with. So the training process took about 10 days to generate this machine over 1 million images or so. And the results of these, you can see, is also doing a good job of localizing, although that was not in the plans. But because the labels were fine-grained, the machine doing the correspondence at a whole image level was still able to locate in relevant regions. So here you can see for a variety of findings, such as hyperaeration, where it is focusing on the lungs, to tubes in the airways, where it is now pointing to an endotracheal tube over here, to looking at sternotomy wires that are found in as post-surgical material over here, and then derivations of enlarged cardiac silhouette, which most people are able to get as well, being in the middle of the chest as such. But you can see this coverage from the variety of findings as well. Now, in the latest work, we have tried to improve this to actually guarantee that you're looking in the right place, and that'll be some upcoming work from our team. So now we turn to the last task, which is to do the report generation as such. So the approaches to report generation, as I said, has been mostly machine language driven approaches. And as I said, this does not guarantee clinical meaningfulness of the report, even though there have been some improvements made in trying to incorporate some knowledge into this generation process. But the latest work that we have done represents the state of the art in terms of the quality of the report that is generated, but even if you use the same blue score or rouge scores for judgment as you go. So the basic methodology is start from a coarse description of a finding, like elevated hemidiaphragm, then go to the next level of classification that describes a finer grain. So it says elevated right hemidiaphragm mild, and from there issue the statement the right hemidiaphragm is mildly dilated. So it's using a combination of deep learning, of both coarse and fine-grained deep learning, using fine-grained labels to begin with, but then combining it with a document retrieval technique to actually generate the report. The overall workflow is like this. You start from a prior set of reports to actually extract fine-grained labels from them and form these fine-grained label pattern vectors called FFL vectors. From there, you back index to the sentences and the report, so you know which finding label came from which sentence and which report. And this set of pre-processing goes into a database so that you have a set of pre-ranked sentences and reports. Now, given a new chest X-ray, you would go through the at inference time, learn and classify the fine-grained labels to form these vectors, and use that to index into this database to retrieve relevant sentences. And then you take the fragments of findings that correspond to the relevant detections within the chest X-ray to reconstruct a report. And that's the overall process that we use. So the overall report generation system has a dual learners, a core finding and a deep finding, and a multimodal fusion module that actually tries to combine the findings followed by a report generation stage. And the details are described over here. And if you take the overall evaluation by putting them to all of these together, both the fine grain and the average AUC for both core finding models and fine grain finding models, you can see as here. From here, you can also see that the fine grain finding detector's accuracy is lower than the core finding detection as expected. But for the majority of the findings, even the fine grain findings maintain decent accuracies. But from the nature of the process, you can see that you can directly emit a label that says things like moderate pleural effusion in the right lower lobe, whereas most classifiers would simply say pleural effusion at best. So severity is assessed as well as laterality and location. So here are some examples of automatically generated report. And you can see that the report that is generated is guaranteed to be natural language ready because we are picking up from prior sentences and reports. However, the methodology of assembling the sentence is focusing on the relevant findings and making sure that these findings are actually likely to come from a report where the joint occurrence of these findings is also likely. And these are the two features that distinguish the quality of the report that we generate. We did an evaluation against a variety of methods, and this was published in MICI last year. So you can see the comparison with different methods in terms of joint text learning or reinforcement learning or template-based retrieval method that have been used. And to date, I think this type of quality is every year the quality will improve, but as of last year, this was the best quality report that could be generated. We have to say that the quality judgment through these lexical or even partially semantic matching scores are not really a good indication of how good the report is from a clinical standpoint. And actually new metrics need to be developed to do the assessment of these as such. Okay, the last section in the few minutes I have left, I will talk about the clinical studies we did to actually assess the performance of automatic report generation, both in the cataloging of the findings and as well as in the evaluation in comparison with clinicians. So the clinical studies we performed are for normal abnormal discrimination as well as for discrete preliminary studies, and the report quality reading, which is a Turing test we gave to the machine versus the radiologist. The normal abnormal discrimination study was something that we conducted in using international data sets, which is trained our model on US data sets, and then tested it in international settings. So this was done under an appropriate IRB. Given the interest of time, I'm going to skip through some of the details, but you can see the main result said that we could filter out as many as 30% of the normals without missing a single abnormal case with the classifier that we built when tested on a brand new field data set as such. So this points to a potential utility of such tools for normal abnormal discrimination and reducing the expediting clinical workflows in some sense. The next study was a discrete label comparison study that we reported in JAMA last year. And
thank you, Dr. Said Mohammed. So we only have 1 minute.
Okay.
Yeah.
Okay. So we can discuss more about these studies. The overall result of it, I will probably say that the average sensitivity and specificity of the machine was comparable to the radiologist from this particular analysis as such. So maybe we'll stop there and leave that as the overall result, that by building carefully these models using a well-known documented process, we were able to reach a comparable performance to radiologists for the particular task that we took. And this is in comparison to radiology residents that showed that the sensitivity and specificity and the positive predictive values are within the ranges of what the radiologists are able to achieve in the automated report generation task that we took on.
Thank you so much for the excellent talk, Dr. Saeed Mohammed. So we also received at least two questions in the question and answer. So let's discuss in the panel discussion. Thank you so much. Our second, thank you. Thank you so much. Our second speaker will be Dr. Le Lü, who is the director at the Ping An Bethesda Research Lab. He has published a lot of papers. So before he joined Ping An, actually he is a researcher at NIH and also the director at NVIDIA AI Infrastructure divisions. He is the IEEE fellow on medical image, AI, and oncology image. He also is a associate editor for PAMI and many other journals. I have to highlight, he also one of the authors of the NIH chest X-ray 14 and NIH Deep Legion. So actually many people use this data sets, include myself. So Dr. Le Lü, welcome to here. Thank you.
Okay, so you can see my screen, right?
Yeah, excellent.
Yeah, today, so I'm actually going to discuss something new. It's not really new, but it's we haven't talked about this. We only starting to talk about this topic like starting this year, but this work has been done in a couple of years. So the purpose of this work is we talk about pancreatic cancer, which is one of the hardest cancer for prognosis. So the basic concept here is trying to show the whole pipeline, from diagnosis to precision prognosis, deep prognosis, and even like a treatment response. So as far as I know, I have been working on. Can I see my? Okay. So this is a couple words we won in last couple of years, and this is our current team with interns. So the topic is really about I gave a showcase about one of the most difficult cancer case and our recent work. As most of the paper was published last year or this year, has not been really present in the conference yet, but you got preview. So this is actually my slides. I give talk several places. So we are at industry, right? Or industry or hospitals. What we do is really for translational medicine. We don't just publish paper for fun. It's really for patient care. Siemens. My first 6 years of my professional career was at Siemens. Then the next 5 years was at NIH. So I'm pretty much like very influenced by Siemens and NIH. So I'm very clinical, relative compared to other industry researchers. I'm more clinical, yeah. So basically, we all think about it is translational medicine. How would we make things that will work in the practice and that will help patients, right? So the three things, basically, this book has been published for more than a decade. It's excellent book about how to make things work, how to make healthcare cheaper and better. So basically, right now, many of the things we do is really intuitive medicine. Means like when I will take examples later on. I will give the overall message at the beginning. Then I use my material to prove that. Right. So right now, still many things. I mean, especially for cancer care, I'm mostly oncology. So for the cancer care is even US it's a lot of like inequality. 85% of the patient they cannot be treated at the top 50 NCI certified cancer centers, right? Even in those cancer centers, it varies a lot, and for certain things, it only can be done maybe by one or two, the top list, like hospitals on the planet, they can do certain things for certain lucky patient or non-lucky patient, right? So really, like what we do is trying to make intuitive medicine that can only be solved by the top expert into the face of empirical medicine. Empirical medicine means like there will be, you may understand, will be a lot of computing, a lot of reproducible quantification process that can be easily translatable, right? Even we want to go to the face of precision medicine, we're not there yet. We're still in the model of the empirical medicine, but it's fine. It's a lot of things you can do in there. So, people talk about the three principles about this book is about how to make things work, especially in healthcare. So people talk about a lot about disruptive medicine, disruptive technology, especially in the Silicon Valley. Really, I think the thing that was something missing was the enablers. What we do can make some nesting happen, some nesting possible, feasible, reproducible, and useful, right? So I give a few examples, and actually 3 examples from our own work, and 1 example from USC, the Nature Communication paper just published last year. So the AI, what AI can do, what deep learning can do is really, like, I think deep learning is really a nesting. I'm not sure, a lot of people, they still, if you are still hesitate or you have question mark, you should not, based on my opinion. So I moved to like NIH in the year of 2012. Right? In the year 2012, I decided to move to NIH. I make it happen in January of 2013. That I remember it was a very cold January. I drove by myself from Pennsylvania back to Maryland. Yeah, the real reason is I feel we can make deep learning work in medicine, in medical imaging per se, and we make it happen. And we did some work at NIH was highly influential, but I think our recent work is more make me more excited because it's very translational. I'll give you 3 examples. So the first work was published in MICAI this year and had not to be published, right? Early accept, and we have a clinical paper that's under review. So basically, we can do osteoporosis using conventional X-ray. So it was not possible. The current protocol is by DXA. And what we do by conventional X-ray is really for opportunistic screening, because a lot of patients actually have such kind of disease, but they were never sufficiently screened. So this disease is underdiagnosed, undertreated, right? So if, imagine all the hospital, you have a lot of X-ray, matter, it's chest X-ray, it's lateral vertebral X-ray, or a hip X-ray, right? And physicians, the top physician, actually not from the radiology department, from the rheumatology department, they can guess if this patient has osteoporosis or not, right? So this domain expert, they can guess. But this thing is not translatable. It's an intuitive medicine. You have to have top expert. They can guess for a few, But we make a computational tool, and we can achieve the quality as good as the hologic DSK machine or a GE DSK machine. So that's the work. The first part of the work to be published in MICAI, and the clinical work is right now under revision. It's not in some. It's already under revision. The second example is actually, to precisely assess the quality or the quantitative score of the liver steatosis, right? So right now, the machine, the clinical practice was done by FibroScan. FibroScan is a special machine, special ultrasound machine, like electrograph, and it has some problem. And basically, our deep learning algorithms, of course, is not easy. It's a lot of work, and we can achieve the quality better than FibroScan by let's say FibroScan's published AUC score is about 0.85, and we are at AUC 0.95. So we're noticeably better than a expensive machine. And also we generate better, we cover better because the elastograph is actually sensitive to the liver inflammation. But our algorithm, our imaging modality and algorithm are more stable against that, right? But this is two topics I would not be, because the time issue. We already partially published, but because of time, we're not going to talk about here. But I will focus on the third, and I will talk about the third. That's using multi-phase CT imaging. We can do precision diagnosis for pancreatic cysts. Pancreatic cysts means like all the tumors, like 10 types of tumors, and the patient management, right? So when we do this, and also I ask you to see the reference down, at the bottom of the slide, that's called the Raptor Night. That's actually, means, what my message trying to convey is deep learning plus conventional imaging modality. They can actually do more than the more expensive, more subjective imaging modality or other means of diagnosis or treatment, treatment planning tools, right? So this work, the work from USC is the HE stain are the most popular stain for the pathology images, and they are very easy to reproduce, right? So for certain of the breast cancer patients, they need to find the receptor or not. If they have a receptor or not, the treatment pathway will be different, right? And right now they have to use the emo therapy, kind of like a stain, like a very expensive, not very reproducible thing to look at the receptor by human readers, right? But the machine or the deep learning algorithm, they can actually just look at the HE stains, the cheaper, the more convenient way. They can achieve the same or even better than human readers in this assessment. They say they want to, basically make a decision if the underlying patient has the receptor gene or not, right? So they actually make it a lot cheaper, right? They can just use the regular stain. They achieve the same result, and for the big hospitals, the cost will be lower, right? The cost will be lower, and the quality, at least not worse than before. Probably is even better. And for other developing countries, you don't even have the method, you don't even have the feasibility of doing this like emo like a pathology stains, right? So and you don't need to do it, right? So this really means like quality up, cost down, and also this has very good reproducibility, right? Because we want to make a medicine cheaper and everywhere, and solve the inequality and solve the cost issue, right? So there are a few examples here. I hope give you a heads up, what's going on. I think it's not hard work, but I think we should have a bright future. Yeah. This is the main topic about, I have been doing oncology for 15 years, and I work with a lot of doctors, a lot of hospitals, and I don't just work with radiology. I work with oncologists and surgeons in different departments as well. So I got a reasonably good overall understanding about the cancer care. This is basically the most popular top 5 tumors or cancers, right? I never worked on brain tumors, but I worked on the rest of the 4 for a long time. And so mostly we do is characterization, right? Segmentation, and the direct from segmentation can do diagnosis, guessing something was going on, or detection, or imaging genomics, means like from pathology images, you can guess what's the genome characteristic of the patient, right? This is just like the example from USC. Or, some people, they feel even from radiology or PET imaging, they can have some strong correlation with the genomics feature of the patient, characteristic of the patient. And more in this work, I actually will discuss more about the prognosis. Prognosis was not really fully solved before. And I think we have some recent work that really pushing forward this field. And prognosis is basically trying to see, hey, this patient can survive for the next 6 months or not, right? Or a year. Within a year, will this patient be still surviving? This kind of thing. And this is based right now is the most experienced doctors can guess, but they certainly they cannot be done better than machine. Certain medical tasks, machines are much better than humans, and they're complementary to humans. They're not going to replace humans. They are make human doctors more powerful in a sense, right? We're not just solving the problem that's easier for human to do, but really something on their wish list, but it's hard to do, and we make it a lot easier to do. So I cannot talk about all the cancers. We work on a few, right? But here, just give you an example is using pancreas. So pancreas in US, the death rate is about the third, right? The number one is lung, the number two is colon. So the number three is pancreas. They have like 25,000 people die in male, and 22 people die for women, right? And this is because the pancreatic cancer has really poor prognosis, right? So the pancreas cancer will be more and more important in a sense, because it's just we actually achieve a lot on managing the breast cancer and the lung cancer, even the prostate cancer. But really, like the pancreatic cancer is one really hard one. We haven't really made much progress in the last 5 to 10 years. And right now, for the best hospital, the 5 years survival rate increased from 9% to about 10%. Right? So in essence, the early diagnosis is very crucial. And if the patient can be early diagnosis, the survival rate will be drastically improved to 25% to 50%. The major thing is because the pancreas cancer, there's basically no symptom at the T1 or T2. Really, like there's no direct symptom. It's not very easy for diagnosis or screening. Right? I will not talk about screening. The screening work is under submission. So here is basically this is basically today the manual. Like I will basically focus on the diagnosis. Forgive me, I have some like Chinese characters in there because I give talk to the Chinese audience as well. Sometimes they cannot figure out what's the English words mean, but I think my English part should be sufficient. Yeah, so I basically talk about these few papers, like 7 papers. This is a 7 papers I'm going to cover. Yeah. So the first work is, I'll go quick because the time issue. I think I don't have much time. I basically show you a lot of pictures. So this is actually the first work that was done like we submitted last March. It was published last year in Mekai. I think this is the first work, multi-institutional first work about solving the sanctation of the PDAC. PDAC is the 85% to 90% of the pancreatic cancer, most deadly one. We probably all know, like Steve Jobs died because the pancreatic cancer wasn't. He actually had one with fairly much better prognosis. Like he actually was diagnosed on PNET, so that's a much slower growing cancer compared to PDAC. PDAC is really like the most aggressive cancer at all. So, the thing is to detect or to just smaller tumors, the really key is sanctation. You have to segment them, or you detect it by sanctation, basically, in a sense. You have to segment your algorithm to figure out where it is, and you can segment a few voxels or maybe more voxels, so you can, you can find the tumor, and you can, give the human reader say, hey, this is potentially a pancreatic tumor, and you need to look at it, or you need to follow up. Even you cannot confirm fully from, let's say, from like a non-contrast CT, you need to do a multi-phase CT, or you need to do some like a MRI or something like it to get confirmation. So this is, we basically, we got data from 4 institutes. This is a comparison to previous work. really we got big data. We got 1,000 patients at that time. Right now we have 6,000 patients. Yeah, we are making progress. And by then, it's a year and 3 months ago, our death number was 0.71. Right now we're 0.76. That's probably the upper bound you can, because even for human readers, their inter-observer variation is about 0.770. Yeah, so I don't know, it's a little bit better than the human readers. And we work with very, very good physicians, just per se. Yeah. So this is basically the technology flow chart. I don't have time to in detail. You can read the paper. I have a reference here. So basically, it's we have student teacher model. We even have a teaching assistant. Basically, we try to gather all the information from elsewhere, everywhere, and even they are annotated in a different level. So basically, we use the technology innovation to bridge the gap, so we can basically learn from whatever. As long as we have data, we can learn. We can improve it. So this is very much from the information theory. You have multi-channel, some channels are noisier than others. But if you are good enough on the fusion, you can always get better, not monotonically improve the result, right? Not to decrease the result. You have to be smart on the algorithm design and really be innovative on solving the problem. But more data are better, yeah, in a sense. So this is basically the data set. I think our strength is we got data set C. That's for a hospital, like we have at the time we have 500 patients. Each patient is 3 scans, so we got about 3,000 scans, 3,000 CT scans, and there's no annotation, but we know the report. So we basically know what patient they have this disease or not. We have their final report, not just the radiology report. We have the clinical report, so we know what's going on about this patient, right? So this is basically, how we get that. This is the first step, and I'm sorry for I only can be able to cover it very briefly, but you can read the paper. The paper has all the details in there. So the next step is a work we are very excited about. Is going to be presented later this month. Will be in CUPR 2021. So we're solving a problem is for pancreatic cyst. Pancreatic cyst here means all the tumors. Pancreatic tumor has 10 types, 10 subtypes, right? 10 subtypes. The PTAX is the dominating one, the most deadly one, but there's some other 9 types of it, right? So the first work in any hospital, when people find a cyst inside the pancreas, the first thing they will ask is this a PTAX or not? So this is basically the first step, all the first diagnosis trying to get, right, for any physician. And all you can see is the pancreatic cyst is actually the prevalence is actually higher than what we originally think, especially is correlated with the age. So if you are, including myself, I'm going to be 50 years old in the next few years. So if you are 50 years old, it's already like a 1.5%, right? So all the people from 60 and above, for Asian is 4.7%. I mean, nearly 5 people out of 100, if your age is about 60 and you're Asian, and the risk is not small, 5%. Yeah. So the best particle is actually from Johns Hopkins. I graduated from Johns Hopkins, but this is my honest assessment, not because I graduated from Johns Hopkins. They are the best. In my understanding, they're the best. Yeah. So our work is very much inspired by their work, by Hopkins' work. They published in Science Translational Medicine in 2019. So it's a very dedicated procedure. Maybe I have here. It's a very dedicated procedure, including a lot of things, a lot of magic things, but they are not really fully translatable or reproducible. Our method is fully translatable because we only need imaging. So basically, we have a sophisticated imaging protocol, and on the top right is our algorithm. So basically, we actually simulate what a human reader will do. We segment the pancreas, we segment the tumor, and we parse the different zones of the pancreas, so we know relatively where is the tumor located, right? We also even know some vessels passing by, some anatomical structures passing by. So when people making decision about what this tumor it is, it's not just how it looks like, also the geometry inside the human body, and that geometry against the pancreas and its surrounding important anatomical structures. So basically, our arm do the same as human readers, just as human reader will do. Yeah. So we have a sophisticated, for this problem, we have a sophisticated task. Sophisticated imaging product, we have 5 phases. So 5 phases is rare, relatively rare, I have to tell you. But it can be done. I mean, when you have a modern CT machine, you can do this. It's just the patient will have more radiation dose. But the current protocol is actually human. The patient need to be need to do a biopsy and doing like a gene screening and the protein screening and the wet labs and also some like a manual reading by the top radiologists on the planet, right? So I think it's still relatively our approach is less harmful for the patient. Yeah. So I don't talk about just that's a geometry. This is our main contribution technology from the technology perspective. It's called SMCN network. This is a new method will be presented in 2 weeks. So basically, what we do is we can significantly increase the detection rate of the non-PDAC. We talk about PDAC is difficult, non-PDAC is even harder. So we basically increase the detection rate from 0.69 to 0.887. Yeah, so it's much better. So we can detect all this like non-PDAC tumors, and we can basically do a differential diagnosis about if this is a PDAC or non-PDAC, or the 3 categories. So 3 categories, what matters most is this tumor is you can discharge the patient, or you have to monitoring them, giving them say, hey, you have to be back in 3 months, 6 months for re-exam, so to see if the tumor grow or not, because they don't know for this type of tumor, they don't know if they will grow into benign, into malignant or not. For the time being, it's benign, but we don't know if that will grow into malignant or not, right? So for the discharge, it has the same probability as no tumor, the chance they will go into malignant. For the other 6, really thing you do, you have to evaluate if the patient can undergo a surgery, because that's other 6 tumor, including PDAC, is you should consider to resect them when you can. Yeah. So this is basically our system on the fly. So on the right is actually the AI will show. AI will segment pancreas, segment the tumor. It will give like decisions, PDAC, non-PDAC, 90% non-PDAC, 10% so non-PDAC, and it recommends surgery. 99% recommends it should go to surgery. Machine is based on a probabilistic classifier, right? So it give a probability. It's not like human will say, hey, is this for sure something? But I think it's typical. It's behind everything is statistics, right? So this is also, we have a small scale human reader study. It's comparable to the best of the expert radiologists focusing on especially some pancreatic disease with more than 10 years experience. So the next step of work, I will go even quicker, is just say, hey, when you find this tumor is recommended for surgery, are they resectable or not, right? This is a hard decision, actually. I mean, really, you want to resect them, but you don't know if they're resectable or not. This is including the treatment recommendation, right? So you have to decide a lot of things pre-operative in order to get the optimal outcome of the overall survival. So this is the work published last year. MICAI, it was one of the best paper candidates, and our journal version has been accepted. We don't know if we will get the best or not. That will be decided in September, October. So our paper is one of the candidates for MICAI 2020 medical imaging analysis best paper candidate. So in that sense, right? So basically, it's a multitask. Work. So we basically predict not just overall survival, but also the surgical margin. The margin has 2 margins. It has 2 values, R1 or R0. R0 means like within the margin, there's no like cancer cells from the pathological images, right? And R1 means like you cannot resect the tumor very well. The tumor already has metastasized, at least within the margin. You can see that there are already some cancer cells. So, or at least some may not be like a remote metastasis, so it has local metastasis in a sense. So of course, with local metastasis, the patient's survival will be worse than without, right? So this is an imaging protocol we use. It's 3-phase. This is very typical. This is a very typical 3-phase. And this before is human gas. This is how human gas. It's varies. It's really like it's art. You have to find the intuitive medicine, the top expert. They can guess better than the other physicians, right? Those physicians not always feasible to find. Just say. So this is basically the network we do. We use this network to kind of do this task on behalf of human. In a sense, this is some like about by that time we only have 200 patients. Right now we have much more than that, and this is the conference version of the statistics. So basically, also the algorithm looking into the tumors versus vessels versus the anatomic structure of the pancreas. The resect or not, it's very much relevant with the vessel because tumor need a vessel to feed in the nutrition they need, and also you have a relationship between with the vessel depending on the cancer is resect or not, basically. Because if they're fully wrapped around the vessel, I mean, it can cause massive bleeding. You actually cannot cut the cancer anymore. Yeah. So this is, I would just, this is basically the prognosis model, and this ROC score, we can predict if the patient is still living, still alive, still, alive in the first year or second year. This is.
Okay, Luis, sorry about this. This 1 more minute.
Okay, yeah. So this is basically, we can divide the patient into a higher risk and lower risk patient, and even the given the default protocol is TNM. That's the typical TNM protocol about making staging about patient, but our algorithm will really make it more personalized. So even within even the TNM staging are the same, the patient actually have different risk. The deep learning or the quantitative computing really make a patient can be more personalized, and this cannot be done by human. This can only be done by computing. I'm not saying just deep learning, but really it should be done by computing per se. Yeah. So this is another example. We not just for pancreatic cancer, we can also using just 3D PET imaging, and we can use for OPSCC. So this work has been published in the CTR this month, and so this is basically showing the methodology of translable. It can deal with 2 cancers, or maybe more than that, and deal with a complete different imaging modality. So this is my last slide. I just played the video. You can have a look about the tumor managing. The tumor managing means the sizing and if they are growing or not. You have to find the multi-time point about a certain tumor, and then you can compare visually, or you can compare them quantitatively. So the thing I talked about here is already like in the process of commercialization. So we basically work with PACS companies. We kind of make this into smart PACS or intelligent PACS for the offering, in that sense, or even some like a research platform. So more physicians they can play around with that. They can use for patients within the hospitals. So that's actually pretty much that's it. Yeah, that's my last slide. Yeah, thank you.
Thank you so much for Dr. Lui for the excellent talk. So for the people, if you have any question, just key in the chat box, and we will have a panel discussion section after world talk. The third speaker will be Professor Lei Xing from Stanford University. He is the director of the Medical Physics Division at Stanford University. He is very famous in the medical image, artificial intelligence in medicine, treatment planning, image-guided intervention, medicine, and application to molecular image in radiology. So he is a PI for many grants like NIH, DOD, NSNA, and other awards. He's a fellow of AAPM and AIMBE, and he is also the associate editor or editorial board in many journals in medical imaging. Thank you so much.
Well, good morning, everybody. So, can you hear me okay?
Yes, excellent.
Okay, great. All right. Thank you. Like to thank also all the organizers for putting together this wonderful conference. So I don't need to give too much introductions. Just want to remind everybody, when we talk about artificial intelligence, we have 5 major topics. And machine learning is pretty much 95% what people are talking about, and most of my talk will be focused on application of machine learning too. So let's start with clinical workflow. I think it's very important, especially for students, to understand, what AI can do and AI cannot do, for our clinical decision making, right? I mean, this is our clinical workflow in radiation oncology. When a cancer patient, or actually we go beyond cancer, we treat a lot of functional disease. So I can briefly mention that. Patient typically goes through a imaging, we call it simulation. The goal here is really to set up a patient model, right? So it's a geometric model that we set up by doing imaging. So this is not a diagnostic scan. Typically we use CT, and occasionally we use MRI, and PET is obviously part of that because you have to identify the target. And then go to the modeling. So this is a part that you build a patient model and then do a treatment planning. So where you simulate your radiation treatment, and in the case of surgery, obviously this is you simulate the surgical procedure. So what is the optimal path for the needle coming in, etc. And then patient go to the treatment room, treat the patient according to the simulation, okay? And then follow up, 6 months and 1 year, 2 years. So there is a follow up. So in this whole workflow, there are a lot of labor, okay? A lot of inefficiency in current practice. And also there are a lot of decision. So on the imaging side, we have to do image reconstruction, right? So these are the task that AI can help us, image reconstruction. So I will come back to this point because the current image reconstruction is not efficient. It's certainly not ideal because, if you want to take an MRI, I'm sure, some people took MRI before, you talk about you staying in a dark, cold tunnel for 40 minutes. So, you need to speed that up, the data acquisition. So modeling, segmentation, right? Tumor target definition, and also registration of different image modality. And this actually, Dr. Lee gave a very nice talk on this topic. And treatment planning. So this is really specific to radiation oncology. So this is a procedure, like I said, you simulate a patient treatment on a computer. And so there is a, the simulation basically comes from 2 parts. One is the geometry, right? You have to select what is the best beam for radiation beams coming in. And the other thing is really the interaction between high energy photon and the tissue. So that is, we have a very accurate model to model that interaction so that we can deliver essentially within 2-3% of the dose that prescribed by the physician. So this procedure need a lot of image guidance and also the patient follow-up, right? You have to tell patient after radiation therapy, what is the predicted toxicity, what is, the survival. So all this decision, can be helped it, or can be made much easier by AI. I mentioned that we treat not only cancer patient, we also treat a lot of functional disease and also even the cardiac, like VT. So this is just an image basically on the heart, and then you actually define a target and then use radiation to treat the disease. So mathematically, I like really to simplify the deep learning into a problem of mapping, right? You mapping from one data domain to another data domain. Really, there's 3 categories. The first one is really you mapping between the same data domain. This is probably the easiest task. Super resolution imaging. I mean, I'm sure you're familiar with that concept, right? That's an example. You just want to really map a low resolution image to a high resolution image. So the second category, the data between input and output belong to different domain, like image reconstruction. So that would be a good example, right? You measure the data and then you map that to image domain. So there the problem is slightly more complex because you have to change the domain. And the third type is really, it's also different domain, but different from second category. The 2 type of data have no physical or geometrical relationship, but they correlate each other, but you don't know what is a relation. Like a clinical trial, right? You gave a certain drug like cisplatin or or immunotherapy to the patient, and what's gonna happen. So you have outcome data. So you try to build a correlative model. So imaging, basically, image is a data. It's actually a very special type of data. Has a spatial correlation. So actually, deep learning has really changed the way that people do image analysis, people do image reconstruction. So these are the commonly used image modality in the clinic. 2D colonoscopy, and then dermoscope, cystoscope, etc. And then on the 3D side, PET, fMRI, MRI, CT, etc. So image reconstruction, this is where actually I can see that deep learning can change the way people do image reconstruction. So currently in all the commercial CT, MR scanner or ultrasound, the reconstruction is based on the physical model, right? Either a model-based or analytical model. So you do a so-called, in the CT, you do a read-on transform to inverse the measurement data to the image domain. So what I'm showing here is a data acquisition process for a cone beam CT. So this is one of our actually a lung patient. And typically you need to collect a lot of data, 1,000 projection from 0 to 360 degree surrounding the patient. So the condition, if you are electrical engineer, you know that you have to satisfy the so-called Shannon-Nyquist theorem, right? Because you have to have enough sampling in order to get a really a decent image. But this is not true anymore when you have a deep learning, because Shannon-Nyquist theorem people call it a no free lunch theorem, right? Because if you want to get a high resolution image, you have to pay. So the reason you have to pay is because you don't include any prior knowledge. And so if once you have a prior knowledge, and then you don't have to pay. So so you can get a free lunch, but maybe you can call it leftover lunch, right? Because it's a free it basically it's a prior knowledge. It's a leftover from the previous patient. So this is conventional image reconstruction scheme. You have a number projection, that's basically sampling. And this is the level of prior knowledge. So if you don't have prior knowledge, you're here. If you have some prior knowledge, and this is so-called the regularized image reconstruction. Before the deep learning, a lot of people work on using compressed sensing, etc., to include some prior knowledge. So you can reduce number projection, but not too much. With deep learning, you can really push the sparsity into the limit. So in this work, we actually push the sparsity to one single projection. So what that means? That means that when you're taking a chest X-ray, you actually can use that information plus prior knowledge to reconstruct a volumetric image. Right? So yeah, this is the reconstruction process. So like I said before, you have a 2D measurement data and goes through, encoder and then dimensionality transformation in the image domain, in the feature domain, and then you do a decoding, so you get an image. So that's how it's a very complex network, but it works. So, and this is actually one of my postdoc, Xiao Kun. So he did this work. You can actually use a fluoroscopic image and then convert that into a so-called a 4D. So this is 4D image. So 4D, 3D spatial and plus 1D time. So this is, you really can push that into limit. So recently, actually, we pushed this reconstruction scheme further. So this is basically try to include, some known knowledge into the deep learning process so that we don't have to have a very complex network, right? Because if you can inform the deep learning algorithm with some geometry information of your imaging system, you can actually do much, much better. So this is already in the archive, you can find it. This is our MR Linac machine. So it's the same thing, sparse reconstruction. You can use that to speed up the MR reconstruction too. So in a nutshell, this is a hybrid between a linear accelerator and MR system. So the MR provides real-time image to guide the delivery of radiation to the patient. But today, so this guidance is actually 2D. So because you have to take a lot of data, so the reconstruction very slow. You can only get real-time 2D image, either sagittal, axial, or any plane, but it's 2D. So with deep learning, you can push that into 3D in real time. Okay. This is another related to reconstruction. So you can actually use a single energy CT, so which is much cheaper and widely available to derive a, derive a dual energy CT, which is very expensive and also has a lot of technical issue. So that this is really a deep learning bring a lot of free lunch to us. And then you can even convert that into a monochromatic energy. So like a 40K, you can use basically a regular CT scanner, get a, monochromatic energy CT. So this is really important for quantitation of imaging modality, but I'm not going to the detail of the application of monochromatic CT. So treatment planning, we were able to again use the treatment plan from prior patients, and then to speed up, to automate the treatment planning process. For a patient like this is a head and neck patient. We talk about 1 day or even 2 days labor of a technician. The only thing he does, is really just sitting in front of the computer and tweaking to get a, desirable dose distribution. This takes 1 day or 2 days for the complex case. And, with the computer, you just with a button push, you can actually get it done. So this obviously has a lot of clinical impact because, reducing. So talk about, AI replacing human. I think this technical repeated work will be replaced by AI. So we talk about the radiation therapy delivery. So this is the machine, has a lot of bells and whistles, but I guess the key issue is how do you visualize, how do you identify the tumor, during the treatment. So I show you the MRI CT machine, but this is actually a integration of X-ray image with a LINAC. So what you're seeing here is a patient, the tumor cannot be seen on the X-ray image. So in order to see them, we have to surgically implant some metallic, like in this case, is a gold, into the tumor in order to see them on the X-ray. Obviously this is costly, invasive. So with deep learning, the question here is, can you actually use deep learning to identify some inherent tissue marker, right? So that you don't have to implant those gold marker. And this is actually, it's being done. You can see that this is a pancreatic tumor that without fiducial, we can actually track the motion during the treatment. This is a prostate patient. So in terms of predicting outcome, predicting the toxicity, this is also very important. Current practice, you basically based on your training, the physician or the resident make a prediction based on some historical data, right? You tell patient you're likely to survive 3 years or 5 years, etc. It's a very inaccurate. So this nomogram-based approach, very inaccurate. It's actually it has a lot of clinical implications. So here with the deep learning, we can train model, including, so it really just individualize the prediction, right? We have not only image, but also the isodose distribution, the treatment plan, and also demographic information. So feed them into a deep learning model, and then you can actually predict, what is the chance for patient to survive, and what are the possible complications. So this is our similar work on colorectal cancer. You can, talk about image guidance. So this is a work in collaboration with the urologist, Joe Liao's group. So we are actually, analyzing cystoscope image. Basically, use the cystoscope to build a existing model, to build a deep learning model to identify the high risk region during the surgical procedure or cystoscope procedure. For those who are not familiar with bladder tumor is actually the most expensive tumor to manage because you know, the false negative rate, false positive, false negative is quite high, and also, you know, the patient has to be, you know, once diagnosed, you know, the patient has to really monitor, you know, almost like every 3-6 months. So that's why it's become very expensive. So using AI, you can actually really reduce the mistake, and then you know, provide some guidance in the OR room. So the same technique actually can be applied to, you know, colonoscopy, right? Colonoscopy is a different problem. You know, it's actually it's a very long procedure. Sometimes you know, physicians get tired during the procedure. So it's very important actually to have an AI just to identify the high risk region. Well, you know, I think one of the very important question is, you know, how do you trust the AI model, right? That's interpretability, robustness, trustworthiness. It's very important. So we have done some work recently along this line, and you know, basically there are 3 type of approach to enhance the robustness, right? So, or to understand a deep learning model. So one category is really you find a relationship, right? You know, like attention, or you know, just a heat map type. So, and the other type is really you know, introduce some perturbation into your network, into the input data, and to see what are the response. So the third type is really, I think it's more fundamental. It's really, you know, how can I understand the feature space data, right? Because right now it's a black box, and you know, once you go to the deep learning model, you don't know, you know, you may or may not get something meaningful out of the model. And I mean, even worse, you know, you get a result, you don't know whether it's right or wrong. So visualization of the feature, I think, is very important. But currently there are many techniques, right? PCA, TSNE, UMAP, etc. So this is actually a very interesting example. So this is a GoogleNet. We used NIH, you know, that X-ray chest X-ray data that Dr. Lee involved. You can see that with TSNE, and this is actually a visualization at different layers. So the layer are here, 32, 69. So with this TSNE, or you know, any existing technique, you actually don't see the cluster. Okay, so this is actually using a learning-based clustering technique. You can see that even at the very early, like a 32, we can see the 14 classes. Right? So that actually go along, you know, when you go, it change from one layer to another. So you can really, you know, monitor this whole process. So I think this type of model is very important, you know, for the future of deep learning modeling, because not only we can see them, we can also tell you which layer is not doing useful job. It's actually making make the model worse. So we can actually prune the network to get a better performance. So with that, I'd like to summarize. Yeah. So I think AI will improve efficiency and quality, and also it will be reduce the cost of healthcare. Because just think about, you know, a human, how expensive, like, you know, like a treatment planning. We hired in our department, we have like 11 technicians just do treatment planning. So each one of them, if they do 30 treatment plan a month, and that's a super dosimetrist already. So, with computer, essentially there's no upper limit. So I think, and of course, the robustness and the interpretability are very important. Also like to thank the student collaborator and also funding agency. And thank you. Yeah, that's it.
Thank you so much, Dr. Singh. Yeah. So for the attendee who have any question to Dr. Singh's talk, so please key in the chat box. So Dr. Huang will organize the panel discussion after the talk. Thank you so much again. Dr. Cooper, so are you here? Okay, yeah, sure. So I can see you. Yeah, our last speaker will be Dr. Lee Cooper, who is right now in joined the Department of Pathology of Northwestern in 2019 as associate professor and director of the computational pathology. Actually, it's really my honor to introduce him because he has graduated from Ohio State University in 2009. So yeah, so go Buckeyes. Actually, he has a very successful career, right? So he's really the role model for in the both the EC Department of Electrical Engineering and Department of Biomedical Informatics. Really my honor. So he joined BMI as a faculty in Emory in 2012. So and have a joint appointments in Caltech, and then joined as the associate professor in the Northwestern. So his research actually funded by the NLM, NCI, NIBIB, and NINDS, and a lot of industry resources. So he published a lot of papers in the computational pathology. So let's welcome again for the Dr. Lee Cooper. Welcome home. Welcome to OSU. Thank you.
Thank you, Ping. I appreciate that nice introduction. It's good to be always with fellow Buckeyes. So yeah, I'm at Northwestern. I'm in a pathology department, although I'm an engineer, and a lot of our work just focuses on building, informatics tools, computational models for prediction, and also, data infrastructure and resources. Today I'm going to just give a background on computational pathology. I think it's a more popular topic, but a lot of people may still not really know much about this, and talk about some of the different analysis paradigms that are used. I'm also going to talk, the main topic today will be focusing on one project. It's really a data engineering project, which is like, how do we collect large amounts of ground truth data? And I'll give, I think, a good motivation for why that's the way to think about a lot of these projects where we have really good deep learning algorithms, and it's really more about getting the right kind of data for them. And then I'll talk a little bit about some of the work we've done in terms of improving the interpretability and the capabilities of some of these commodity deep learning models that are used for things like object detection or classification. How do we really make those more specific and function better for pathology? So, there's been a lot of success. I think, when I first started messing around with this stuff, it was just kind of a toy or a novelty. But, starting around, 2013, 2014, there started to be some really interesting results in applying, deep learning algorithms to medical images and in pathology in particular. So, there was a study that showed that, using this data set called Chameleon, that, deep learning algorithms can surpass humans in detecting metastases in breast cancers. In lymph node sections when you put time constraints on the humans. So, in practice and clinical practice, time constraints are a reality. And so, that was, I think, a big moment in our field to realize that we could, achieve similar performance to humans and perhaps even, exceed their performance. There are a couple companies, PathAI and PageAI, that each have, over 100 million in Series C funding. It's really a tremendous accomplishment. Page had published a really nice paper in Nature Medicine that showed that if you get these large data sets where you have these massive images and you have like patient level labels about whether someone has a cancer diagnosis, you can really train without using any kind of annotation and develop really powerful diagnostic systems. And there's also work that looks at predicting things like mutation status from histology. It's not extremely accurate. It's definitely not going to replace sequencing, which is getting cheaper and cheaper. But it's an interesting result, and it could be used for sort of screening for eligibility for, applying some of these assays. So, computational pathology, what's really the potential? I think, the most exciting thing is that we could take sort of the expertise of the best pathologists in the world and, infuse that expertise into these algorithms, and then make that quality of diagnosis available to everybody, whether they're in, rural Ohio or Illinois, or in our major cities where they go to the James or where they go to our cancer center, or out in lower and middle income countries. We can package this expertise up in algorithms and then disseminate it out to the world. And I think that would really be a tremendous accomplishment. Specialization and experience matters when it comes to diagnosis, and that is a limited resource and not available everywhere. So let's try to make that available. All of the rules that pathologists use to diagnose diseases, they're sort of done through, studies, but they're, subjective and kind of empirical. And so what we could do is to use algorithms to generate, quantitative measurements from histology, and then use those measurements to inform these diagnostic criteria. So I think that's another big opportunity. Improving the work lives of pathologists, they're really, mostly overwhelmed. I think there's a big decline in the number of pathologists that are training in the United States, but there's increasing demand due to, cancer incidence and aging population, et cetera. And so it'd be really great to be able to provide them with some tools that allow them to, handle a larger volume of cases and sort of, ignore the easy things and focus on the more interesting or more challenging cases. And then finally, I think, a big opportunity is not in diagnosis, but in how we could use these tools to characterize tissues, sort of like omics, but with images, and make that really a research resource where it's a commodity that people can use. And that could really advance our understanding of biology and disease. And so that's a big motivation for the type of approach I'm going to discuss today. So one of the paradigms is what we would call, end-to-end models for whole slide image analysis. So this is like the two papers I mentioned before, where you have kind of slide level labels like cancer diagnosis, yes or no, or mutation status. And then, you consume these massive images and learn and make some predictions, by consuming, billions of pixels for each sample. These are really powerful models. That's really exciting. It's kind of like really follows the deep learning paradigm of like raw data in and predictions out. It's just like pure machine learning. And I think, it's a really powerful approach, but there's some, I think, challenges to using these types of techniques in some diagnostic applications and definitely in research. So, if you're doing cancer diagnosis, it may be okay to have these types of models that are black boxes, right? A model that looks at a prostate biopsy and just highlights the regions of cancer and tells the doctor to look at those areas. That's fine. You don't really need to interpret those models. But there are some cases where interpretability, might be necessary, or where you might need a different approach. And so a lot of people have applied sort of interpretability methods in these whole slide imaging approaches. They're really post hoc, and so there are challenges with doing that kind of post hoc interpretability. The first thing is falsifiability. So if you have like a heat map of an algorithm and where it's looking, you can't really prove that wrong in any sense. And you can always, it's kind of like horoscopes. Whatever interpretability research you do, you can always come up with some interesting pattern that it's found, but it's still essentially a black box. So an alternative approach is something called dense mapping. And so this term is from a paper that came out in March from the Path AI group, where it's in Nature Communications, where they generated all these annotations. And what they do is essentially map all of the tissue structures and cells, classifying them and segmenting them, with, terms and class labels that pathologists understand and appreciate. And so this is really kind of driven by like annotation of images. And, we have similar work that we've published, back in 2019, we released a data set of breast cancer segmentation where we show, if you get people together and trace, breast cancer tissue regions, you can use kind of commodity models for semantic segmentation and get really high accuracy. And a paper we released more recently on arXiv just this year, looking at the same problem, but with nuclei. And so this dense mapping is a different paradigm. It's really kind of more like bottom up. So we use the deep learning algorithms to really accurately detect, classify, and segment. But we're starting from a point where, we're working with concepts and things that have semantic meaning that, the pathologists already work with and are familiar with. And so it's less of a black box. So, an example of the type of thing you can do with this, we work with this International Immuno-Oncology Biomarker Working Group. And what they do is they come up with rules for scoring immune infiltrates in breast cancer. So if you look at solid tumors and you look at the immune cells in there, for many solid tumors, the type and the quantity and abundance of immune cells is predictive of outcome, independent of other things. And so, you can take these sort of rules that humans have developed and you can encode them into quantitative systems using this kind of dense mapping approach. And so our student, Mohammed Amgad, who's done a lot of the work that I'll show you today, has done some work with this immuno-oncology working group, but also looking at, how do we build these algorithms and score things like, immune infiltrates in breast cancer, and just showing that you can really make this highly reproducible. And you can do this in a way that is consistent with breast cancer experts scoring these same tissues. So these mapping approaches, I think the thing that's exciting about them is they enable discovery and translation. And so what we do is we generate all these annotations, and then we just leverage algorithms from the computer vision community for segmentation, detection, and classification. And so the algorithms that are out there work quite well. I'm really less kind of interested in the algorithms. I'm more interested in how we produce the data. And if we can generate these mappings of tissue structures and cells, then you can sort of come up with quantitative but really interpretable measurements of tissue morphology, things that make sense to pathologists. And what that allows you to do is to sort of test specific hypotheses, and you can't really do that with the end-to-end deep learning models. So for example, you could look at like stromal cells in breast cancer and look at their morphology and some of the patterns and look at relationships between, genomic data or clinical outcomes and response to treatment. That's essentially, impossible to do with these sort of end-to-end models. They don't really have any, notion of different cell types that are encoded in them, right? Of course, interpretability is important when we talk about, clinical translation. I think some of the more immediate opportunities are to use these quantitative models and approaches for dense mapping to measure characteristics that pathologists can see and appreciate, and to identify which new patterns, still add predictive value to the current grading practices. And then we can take these and, try to translate them back into clinical practice by teaching the people how to score these things. So you have, in that case, the loop of the humans generating annotations to produce the algorithms, and the algorithms being used to identify biomarkers, and then translating those biomarkers back to the clinicians and teaching them how to score those things. So this is a post from Andrew Eng recently that I was really glad to see. I think it kind of illustrates our idea about, how this research should go. He's talking about how can we develop new challenges that focus more on data than on code. And I think this is it's great to see someone with the stature kind of advocate for this. And so the idea was to have like a challenge where the code is fixed and you focus on engineering the data, right? And so when we're talking about this dense mapping approach, it's really about engineering the data and how do we create the data and make it high quality. And I think, as the algorithms work advances farther and farther, and the algorithms that are just sort of off the shelf are very good, there's going to be a lot more intense focus on data. And I think that really aligns kind of with our academic mission, because we have the hospital and we have the experts here. Okay, so this is an example of what you can do if you can collect massive amounts of data. So this is a study we published in Bioinformatics. It looks at crowdsourcing breast cancer tissue regions. And so we had people annotate 50,000 tissue regions, tracing essentially boundaries. I'll describe how we did that later. But what you can do if you can generate this data is to make extremely accurate computational models using things just like MaskRCNN, just like regular semantic segmentation models. So what I'm showing you on the left here, these are actually segmentations generated from a computer. These are on cases from medical labs that were held out of the training set. So these are from labs that have not been seen by the algorithm before. And it's really pretty accurate. If you look at this, it's sort of hard to tell whether this is done by a computer or a human being. the computer tends to be a little more detail oriented and less kind of lazy than the people. And so that's maybe a little bit of a tell, but we're getting, extremely high accuracy from these algorithms. And the secret ingredient really is the data. It's not this specific approach or the network that we used. So I'm going to talk now about a protocol we developed for annotating cell nuclei. I won't bore you with like breast cancer biology, but there are, a lot of different types of cell types you could be interested in. We looked at, at least 12 cell types and did our annotations. And so these annotations were performed by a group of non-pathologists, so like medical students. And then we had pathology residents, pathology fellows, and pathologists. And so they sort of work together in this group with the medical students doing the bulk of the work, okay? So, we collected over 220,000 annotated cell nuclei. And then we defined these groups. So we have the raw classes that were annotated. This includes like all different kinds of cell types, some that are very uncommon, like these eosinophils, for example. And then when we're doing our analysis, we're essentially grouping these into different classes, tumor cells, cells you find in the stroma, immune cells, and other types of cells. So we call those things superclasses. So the mechanics of the annotation are that we have a weak algorithm that goes in and makes suggestions for the humans. So it's a very weak segmentation algorithm that just does a kind of a decent job, but not good enough at segmenting nuclei. It just uses basic image analysis, like primitive operations. And then we use rules based on morphology to classify things into different cell types. And so that's just an example of what the human beings will see when they're doing the annotation work. We also provide suggestions from region annotations from our data set we developed previously. And then what the volunteers do is they go in and they confirm by clicking correctly segmented cells. So that just involves a single click for each of those cells. For cells that are missed or incorrectly segmented, what they'll do is they'll draw a bounding box. And so what this allows us to do is to collect a combination of segmentation data and bounding box data, which we call a hybrid data set. And it really relieves the annotator from having to actually trace individual cells. So we get that kind of segmentation data for free. And then once we clean up this data, we get rid of the bad segmentations, and we have a mixture of, like I said, these good segmentations, polygons, and bounding boxes. So one of the things that we did that I think was interesting was to use, pardon me, was to use this mask-R-CNN algorithm to take our like weak segmentations that we use for suggestions and to improve the quality of them quite a bit. And so you can think of this as like fitting a line to noisy data. So we have all these noisy segmentations using just like basic image analysis operations. And then we feed them into this deep learning model, which is essentially just like a curve fitting algorithm that operates, as a function approximator. And just by doing that process, it really tends to improve the quality of the suggestions we show people. So you can see like in the center here, we go from, a bunch of segmentations that are sort of missing these tumor cells in the lower right corner. And once we pass things through the mask-R-CNN and train it and then use inference on the training data again, we get these nice tumor cells that show up and also this fibroblast that was missed. So this is something I haven't seen before. I think it's some novelty that we have, which is to sort of bootstrap our way into getting these kind of higher quality suggestions that we have. So we have two types of data sets. One is a single rater data set. This is where we take a bunch of ROIs and we assign them out to different annotators. And so the purpose of this is to generate breadth. So we have like the scale of data that we need. In this example, all of the people who are junior, who are shown in pink here, they have a Slack workspace where they can ask questions for the residents and the fellows and pathologists, and they get feedback. And then at the end, all their annotations undergo like a quality review before they enter into the data set. Then we have a multi-rater data set, which essentially is used for looking at inter-rater variability. And so what we do in this multi-rater data set is we show them different combinations of things. So we show them, first of all, this unbiased data set where there are no suggestions. So we just get their bounding boxes on everything. We don't really bias them in any way. And there's a washout period, and we show them the noisy segmentations that are the sort of the weak algorithm suggestions, and another washout. And then we show them the refined suggestions that are generated by mask-R-CNN. So this sort of study design allows us to look at differences between people and also the impact of the suggestions on the annotators as well. So one of the challenges is when you're looking at inter-rater concordance, when you're talking about cell detection, you know, it's much more challenging because you can't like compare pixel to pixel like masks. You have to figure out correspondences between, you know, different cells that have been annotated by different people in the same area. And so that required us to generate a new type of constrained clustering algorithm. I don't want to go into this, it's like a little too complex, but it just kind of illustrates the point about, you know, challenges in simple things like measuring inter-rater variability in pathology. So what we did was to, you know, create this clustering algorithm that allows us to essentially generate a type of ground truth from our pathologists. And we also use a EM algorithm to kind of estimate, you know, a single ground truth from pathologists that we can use to evaluate performance. And this essentially just like weights pathologists by class to figure out who's good at what, who's more sensitive, for example, with tumor cells, and who's more sensitive with stromal cells, or vice versa. Okay, let's talk about the data set itself. So when we started looking at some of the statistics, there's some interesting patterns that emerge. The first thing we looked at is detection accuracy, and you see there's really like a range of precision and recall for different people. So we have a few people who are kind of outliers, who are just not really good, you know, annotators, but most people sort of fall along this curve of being like either having better recall or better precision, which is what we typically see from algorithms or these types of inter-observer studies. One of the things that's interesting is that our junior pathologists tend to have a little bit better recall than the senior pathologists. One of the hypotheses we had in this case was that maybe they have more time to do this, and so they tend to spend more time, you know, annotating and looking for missing cells, where you know, maybe the senior pathologists just kind of go a little quicker and don't pick up on those things. But we have, you know, pretty good, you know, I would say for compared to our ground truth, we have pretty good accuracy for detection. For classification, it's pretty similar. You know, when we look at sort of the superclasses and our macro averages, you know, we get a pretty high AUC. Okay. One of the things we looked at then is the impact of these suggestions on people. And what you really want to do is to look at, you know, the suggestions essentially bias people in a negative way. And the second question is, how much time are the suggestions saving? So one of the things we looked at is, you know, for this acceptance rate for the suggestions, how does it vary whether we're looking at our noisy bootstrap data versus our evaluation data set? And so quantitatively, we can see that as we go through this MASCAR CNN refinement, people are clicking more of the segmentations because they like them better and they're more higher quality. And you can also see that in the figure on the left over on figure A. If you compare the middle column of figures versus the right column, you can see that on this refined data set where we've applied MASCAR CNN, you get like a lot more clicks. And so that essentially saves people time. Okay, so let me skip ahead here. So let's talk about the modeling and interpretation component. So we have this data set with 220,000 annotated nuclei. The next step is to figure out, okay, how do we model this and generate algorithms from this? And one of the first things we looked at is like kind of the differences between these nuclei data sets and data sets like COCO that are used to develop like faster RCNN and all these detection algorithms. And there are really some differences in the statistics, like in terms of the size of the objects, which is going to impact like network design and kernel size, but also the number of objects that you find in a single image. So if you look at MS COCO, it's got, you know, pictures of natural scenery and stuff like that. You see roughly 7.7 objects per image. That's kind of the average in COCO. And then I think the average size of one of these objects is like, it takes up roughly like 10% of the image, okay? When we're talking about nuclear data sets, and ours in particular, we average 34 objects per image. And the average, I think, size of something in the image is like 1% or less. So the objects are smaller, they're more numerous. And that should really, I think, you know, inform the algorithm design. So we did take this MASCAR CNN algorithm and then adapted it with these things in mind. one of the things that we looked at is making sure that we can handle variable sizes of images without like resizing everything to the same aspect ratio, or I'm sorry, to the same fixed size. This really helps with like, avoiding distortions of the image content and your aspect ratios, which is a bad thing. Another thing this model allows us to do is to look at training with hybrid data sets, so a combination of bounding boxes and boundaries. That's, I think, a first thing that we've seen, so normally you either have to work with bounding boxes or with boundaries, and you can't really combine both of them. So this network combines both, and it kind of does it through this masking process. So we don't train the segmentation loss if we have a bounding box. If we have a segmentation instead of a bounding box, then we train that loss. So another thing we did is to increase the independence between the classification and detection branches. This doesn't really help with performance, but I think it really helps with like freedom in designing the algorithm, and it gives you more flexibility and allows you to develop an algorithm that works faster, and it makes the tuning much, much easier. So. Okay, so here's some results from our algorithm. So overall, our median average precision was 75. That's pretty good. Segmentation DICE for the nuclei, it's 88.5. That's pretty high for DICE. I mean, it's not like some of the things you see in the tumor segmentation studies for like radiology, but I think it's pretty good for pathology. And our classification ROC was 86. So one of the things to note is, I'm showing sort of two rows of results here. On the top of one of these rows is the prediction, and then on the bottom is the ground truth. And one of the interesting things is that where the algorithm and the human disagree, the algorithm is not always wrong. So there are a lot of cases where we see the algorithm will pick something out, and then that doesn't exist in the ground truth. And so you'll see it'll be called a false positive, for example. So right here, for example, you have these stromal cells that have been detected by our algorithm. Those were missed by our human annotators. They just weren't annotated for some reason. So this sort of just points out, I think, the kind of fuzziness and like evaluating the performance of algorithms in this area. No matter how hard you work to collect ground truth, there's going to be imperfections and errors in it. So there's always just some kind of gray area. But overall, we were, satisfied with the performance of these. And I'll note, we made just minor changes to this mask RCNN algorithm. This is not like a, cutting edge segmentation algorithm. It's just sort of what everyone's using. I think the key to getting this kind of performance is really just like having a large amount of training data, on a bunch of different hospitals, right? Okay. So the next piece that we worked on was like, if we have for example, a nuclear classification algorithm, how can we develop a process that will allow us to take this black box algorithm and understand some of the rationale behind its predictions? So what we came up with was this idea of generating a companion algorithm. So we have this classification algorithm, and we have a companion algorithm that is not deep learning based, it's tree based, and it sort of models the behavior of the deep learning algorithm, including all of its imperfections. And so the way that we did this was we have our segmentation data. From our segmentations, we can calculate all kinds of morphologic features like texture, shape, and size. And then we use those to train this decision tree algorithm to approximate the performance of the deep learning classification algorithm. And then what that gives us is this tree that has all kinds of decisions based on morphologic features that we understand. And so that allows us to sort of like try to understand what types of morphologic cues this deep learning algorithm is using for classification. So I'll just skip ahead here and show you this example. So we created this decision tree, and then at each point in the split, you see a feature that's used in making a decision between different types of nuclei. So at the top level split, we have the difference between immune cells and tumor cells. And what's being used there by the algorithm, the primary determinant is major axis length. So if you have a long major axis length, you're more likely to be a tumor cell or a stromal cell. If you have a short axis length, major axis length, you're more likely to be an immune cell. And that's true. Immune cells are kind of like small and round. So if you go down the left side of the branch, the difference between, say, plasma cells and lymphocytes is really kind of based on the intensity of the staining. And so plasma cells have kind of like a lighter and rougher texture. The lymphocytes are really small and dark. And so we see that, we see that pattern here reflected in this tree. If you go down the right branch of this tree, what you see is that, some of the other differences in terms of determining tumor versus stroma are the minor axis length. So tumor cells tend to be like just kind of large and oblong, where these fibroblasts and stromal cells will tend to be like really kind of more spindle-like. And so the minor axis length is really what's being used to distinguish tumor cells versus stromal cells. So this type of approach, I think it can be used with any kind of supervised learning. And it just lets you sort of model a deep learning algorithm with a tree, which I think is kind of a neat approach. So just to conclude, I wanted to acknowledge some of the people who participated. Mohammed Amgad, of course, who conducted all the study and managed the efforts of over 30 volunteers. All of our volunteers, many of which have gone on to pathology residency. We really appreciate their help. And our team, who also worked on the annotation platform that we use, as well as the funding source and the people who pay us to do this. And then finally, just to point out, this data set is all completely open. Our region data set is also open. We have a web page we set up where we describe the entire process in generating this data. There are videos that walk you through what the data is and how it's organized. And you're free to come and critique this and use it to benchmark your algorithms and to develop new algorithms. We would really like to see that. So that's my time. Thank you.
Thank you so much, Dr. Cooper. Yes, it's excellent talk in the pathology. Yeah. So we are conclude this section 4, and again, so thank you the 4 speakers for the excellent talk. For the next 15 minutes, I will invite my co-organizer, Dr. Kun Huang from the Indiana University, to host the panel discussion with the 4 speakers. Actually, I would like to thank all the speakers. They already addressed many questions in the question and answer sections. So Dr. Huang, please lead the discussion for the remaining questions. Thank you.
Yeah. So I mean, there is one new question coming up in the Q&A panel. Actually, that's for Dr. Lee. That is talking about getting ground truth. Could be very tricky, and just could Dr. Cooper, I'm sorry, Dr. Lee Cooper, and to about if Dr. Cooper can elaborate more on the clustering method. Yeah.
Yeah. So I'm going to refer you to our paper, but I agree with you. It's very tricky, and I think when we did our segmentation project first, it was more simple. We can just use like the same metrics people use in radiology, things like DICE. But when you're talking about discrete objects, there's a process of mapping. So if you have 8 people looking at a single tissue area and they each have 8 opinions, you have to kind of figure out, like, for a given cell, are they talking about the same cell or different cells? And so that's really where the clustering is necessary. So I think if you take a look at our paper, you'll see how we address that. But I think that's a big problem for pathology, is dealing with those discrete objects, because there are many of them.
There is another question. Actually, it's the same question I would like to ask actually for all the panelists. And because you guys gave us such a wonderful talk today, and with a clearly translational value and the translational applications. So then the question actually I think I have, but also the audience also have, is what are the next steps, and how do we really translate this into clinical practice or clinical workflow? So, I mean, since this is a common question, I think I'll just start with, I'll ask all the panels members. So I'll start with Tanvir, Dr. Saida Mahmoud.
Okay, cool. I will answer that. I was also wanting to say that in terms of the ground truth question, there was an additional point I wanted to make, if I could briefly comment on that before I get to this question. So one of the methodologies, we had the difficulty that each image would have multiple findings, and we had to make sure all the findings were covered in the ground truth process, right? So we use a triple consensus with adjudication as the methodology, which is reasonably well accepted in clinical literature to do that. And this requires having specialists with sufficient training and those sort of filtering. but having them do them independently and then come and to get together in a room and hash it out until they are satisfied with the consensus they have achieved. So this was in discussion adjudication process that you can do for, I mean, we were able to do that for about 3,000 images, but the entire triple consensus adjudication process took us about 6 to 9 months to achieve for 3,000 images. So this can give you a sense of how difficult achieving gold standard ground truth data set problem is when you have multiple findings simultaneously present in one image. Now, coming back to your question of, how to translate into clinical practice. So one of the important things is to do clinical studies. I didn't get to fully describe these in my portion of the talk as we ran out of time, but doing clinical studies is very important as part of the staging for this. Now, one of the difficulties from a regulatory standpoint is to do this clinical studies, keeping the regulatory oversight in mind, like what would the FDA regulatory process would require, and doing the studies, keeping those things in mind, which also means that the software itself has to be produced using a QMS or those sort of operations. But once you're able to do that, you can conduct pilot studies. So one of the studies we've done is one such pilot study where you cannot implement it in clinical practice, but it has to be a parallel workflow. So the translational aspects of it go, first is a soft introduction, where you can do this as not impacting clinical practice, but observation in a prospective fashion. And then going into adoption phase if you are going for commercial, then you have to follow the FDA regulatory process. But if your institution wants to adopt it for their own private use, it may use a different process as well. So the translation to clinical practice certainly needs full-scale evaluation before you get to the pilot studies, and then you go through the pilot studies as necessary for getting a regulatory oversight. I can tell you that FDA regulatory paperwork for AI algorithms has simplified. However, they still don't have a good way of treating multiple findings. When the number of findings is in the hundreds, they just don't have the machinery to approve such things at the moment. So this is one of the great difficulties in scaling AI algorithms, when the machine learning models are able to recognize large number of findings simultaneously, but the evaluation and the methodology for approving them for pre-market clearance is very difficult.
Great. So Dr. Liu, do you have got any comments?
Yeah, so I have a humble understanding. Like, I work at Siemens. We build FDA clearance stuff, right? And when I was at NIH, we had meetings with FDA folks. They're actually doing research. They're not doing the approval work, but they are the backbone. They're doing research on deciding what can be approved, what cannot be approved, in a sense, right? So they are doing kind of like counter research in a sense to figure out what real means, what's really significant or not. So my hypothesis may not be correct, but I think the FDA clearance does not mean you are really helpful for the clinics. Honestly, I think that from my own experience in Siemens, we do FDA clearance stuff, but I do not feel that's really extremely helpful. So in my understanding, the FDA clearance standard is between, you have to be safe, right? You don't hurt patients, but it's below being very clinically useful. That's my assessment. You have a lot of FDA clearance stuff. They don't really sell well. They don't really help as you expected. I think FDA clearance is one factor of doing certain things, a necessary condition, but it's not the all thing you need to achieve in order to be clinically useful. The goal, at least I think we should achieve, is clinically useful. They need help patients, help physicians. They need to make the way they can pay. They want to pay for the models or product you are developing, because only if they are willing to pay means you really make a value. I think that's actually above the FDA clearance standard. Of course, eventually we want something is they cannot live without it, right? That's even one bar above. I think FDA clearance is nice, but it's not the really end of the story. You need to do more things, way more things beyond that. So, and also, one thing, at least at old days, is like once you FDA cleanse some stuff, the thing need to be frozen, right? And at least before. Like at Siemens, let's say we have an old version, which was FDA clearanced, but we have a newer one, which is way more better, but we cannot afford to spend the next couple $1 million to do it again. That's I don't know what to say. It's kind of silly, but that's not just we need to work on. I think FDA need also looking into this issue, how to make this thing work, how to make something translatable. Somewhere they need to be less rigid, and I think somewhere they need to be more rigid. In a sense, we all need adjustment. In a sense, otherwise, it took too long. It takes too long to see patients. I work in actually that completely changed my life. I saw this. I'm mostly oncology. I saw many patients on the hallway and actually like really suffering. And of course, they already at the best care ever possible on the planet, but I mean, really, it's limited patient you can care anyway. So that's really the best of the intuitive medicine and empirical medicine on the planet. But we need more than that. Just saying. So right now, our strategy is I want to first make sure our things are really, really clinically useful before we go to FDA, before we go to the clearance. That's something we want to make us. Want to have a scalable way to do an even larger clinical trials, working with more hospitals, even open platform. We dockerize the stuff we build. We work with PACS companies; they can help us to deploy. So really, like a more hospital can be used. I want to. Hopefully, I'm working for a company. I don't make all the decisions, right? But just think from a researcher perspective, or from a person I work with physician every day for the last 8 years. Really, every day. I have a meeting with doctor every day, Monday to Friday. I think we really may want to make sure something really, really useful. Then we go to FDA clearance. Then we deploy to more places for money. Yeah, that's basically what I want to say. Yeah, thank you.
Great, thanks a lot. So Dr. Singh?
Yeah, well, this is a very important question. I mean, obviously, we're working to change the healthcare. In my opinion, I feel there are 2 ways to translate your research into clinical practice. If you're working in a clinical department, like me, or if you are, having a very close collaborator in the clinical department, which I strongly recommend you do it if you don't have one. It's possible, and we actually did translate something into our own practice, okay? Because that's so there is a big difference, right? Obviously, if you do it in your own clinic, that impact is not huge, but it's a first step. So I think it's an important step, being able to test it in the clinical practice. I think the key issue is really the workflow. I mean, just being able to, say, increase the dice coefficient by by a certain percentage, it's not enough. You have to ask the question, at what cost, right? Because these days in the clinical environment, there are a lot of restriction. I have to say that even you installing a software on a computer in the clinic, you have to go through the IT. You have to, you just have to go through a long process. It may take longer than to get that work in the clinic, then you spend time do your own research. So sometimes you have to balance. You have to basically say, hey, is it worth for me to really go through all the hassle to make it work in the clinic? Because you say, maybe I can spend my time better to do more interesting research, and then let other company to take over and to translate into the practice. And I think it's all, in the end, it's all, you know, a trade-off. So the other path is really license your technology to company. We were successful on that. You know, in some technology actually get licensed through the university to some companies, and they are obviously professional, and so they can make the software, you know, robust and then industry grade, and then they have the marketing team, or, you know, really to sales team to really, you know, penetrate to the individual hospital, because that's not a trivial. I mean, I think I would really, if it's not a huge technology, and maybe that's a good way to go, right? Just license to somebody who is good at it, and let them to finish the next few miles. I mean, really, I think those are the important things that the people need to keep in mind. You know, licensing is actually very useful.
That's great. That's a wonderful point. So Lee, do you have any comments?
Yeah,
I know you also work in clinical department. So
right, I think the opportunity is in clinical trials, right? And I think the first thing to do is just to sort of build the operational capabilities to do those things, which is really challenging in academics, right? So, you know, before you use the predictions to influence any kind of treatment, I think you need to show that you can generate them and provide them to, you know, the clinicians in a, you know, quick manner that, you know, where they appreciate them and they can visualize them. And that's not trivial. So a lot of those operational pieces, I think, you know, it's hard to accomplish in academics because we have like postdocs and, you know, graduate students, and they're just not really made for, you know, doing those kinds of things and building like reliable systems. So, you know, it's interesting because I think in academics, you know, we have the proving ground for these algorithms. You know, we have our own research, so we can, you know, quickly develop things, and we're very agile. But a lot of the stuff we need is what's found in industry. And so, you know, I like the comments about collaborating with industry for licensing. But I think, you know, we need something even more agile than that, where we can sort of, you know, cooperate with industry in other ways to advance this field. So I don't know what the answer is to that, but that's what I would like to see, is to be able to leverage more of their resources that we need to do these types of studies.
okay, wonderful, wonderful. So really thank you all the speakers for your wonderful comments and the wonderful talks. So we still have some questions, but our time is running up. So I guess we'll just leave the Zoom open if there is any questions, but I think the organizers may announce something about lunch break.
Yeah, thank you so much. We don't have anything to announce here. I just want to show some comment. We have a lot of great comment in today's section, like the invited speakers in this section are among the top researchers in the medical image, and we have a great conference, great motivation. I just want to show this excitement from the audience to you. So thank you, everyone. Thank you for the speakers. I really appreciate you attending this meeting. Thank you.
Thank you.
Okay,
Thank you.
Okay, thank you very much.
Thank you for inviting us. Yeah, thank you.
Yeah,
Thank you so much.
Yeah.
Okay.
Bye.
Have a great day.
Bye-bye.
Bye-bye.
Bye-bye.